Once again, I should thank the organizer for letting me to speak at this conference. So this is a great conference, great talks, great science. So hopefully, as the last guy, I am hoping I wouldn't break this record, or wouldn't break this nice pattern. So today, the talk want to introduce a concept called cross-species co-expression modules. So this is an ongoing analysis. So I am very happy to take suggestions from you guys. So probably on our way to the airport. Okay. So I guess we are all familiar with the idea of doing gene clustering analysis. So given the genome-wide expression profile, namely you have the system-wide expression value of every gene under a variety of conditions, you can plug it into this clustering algorithm and arrive at some modules, or people call functional modules or clusters. So there are literally tons of algorithms doing this kind of work. So because ranging from some naive hierarchical clustering to a more sophisticated network-inspired algorithms. So the essential idea is just to look at genes with similar pattern across different conditions. So because when genes are co-expressed, they might be regulated by common transcription factors, or they may be taking place in the same pathway, or they might be forming a complex. At the end of the day, they look like doing something the same in a biological context. However, given if you have the expression profile for two species, so what will happen? Probably what can we do is we do it separately, and then at the end of the day, we compare them and try to make a story. So the key message of this talk is, can we do it in a more integrated fashion? So what we want to do is to incorporate the idea of orthogonal pairs between species into this clustering analysis. So we use a common viewpoint in evolutionary biology, saying that a pair of orthologs should responsible for the same function across two species. Okay. So using this assumption together with original assumption that co-expressed genes should be responsible for the same function in a species, so we kind of join these two assumptions together to characterize function in a cross-species sense. So at the end of the day, the modules are no longer independent, but in the form of something like a Venn diagram. So in this module, you consider a set of genes from species A and a set of genes from species B. And they overlap each other in the sense that they might have they are forming orthologous pairs. So the essence of this whole integration kind of framework is a spin model, or more technically, is a couple post model. This kind of model I usually study in statistical physics. So I want to use a toy example to illustrate the idea behind this algorithm. So here we have two expression networks, one from species A and the other from B. So the co-expression networks labeled by the blue links relate nodes from species, so within a species. And then there's another type of links, this green one, which connect a pair of orthologs. So this guy and this guy basically is an orthologs between these two species. So, so we assign spin value to all these nodes. So at the end of the day, the spin value are basically the labels of the modules. So for example, in terms of the output, all the nodes with spin value equal to 1 will belongs to module number 1. So of course, there are in the order of n to the power q different ways to assign this spin value to these nodes. So we define an energy function to characterize these configurations. So the idea, so there are three terms in this energy function, namely these two basically refer to the co-expression network, while the last term refers to the orthologous pairs. So let me try to walk you through. So this W is nothing but the adjacency matrix, +1 or 0. So if a pair of nodes connected by blue links, they are really in the same value. Say these two nodes connected with a spin value equal to 4, this delta function will be equal to 1. And therefore, this term this pair will contribute to a negative 1 term here, which means the energy is lower. So there's a term Pij here, which can regard as a correction, in the sense that every pairs are corrected by somehow different values. These corrections are designed so that nodes with different degrees can get a different reward or punishment according to these spin values. So if two nodes which are in the same value, but they are not connected by the blue edges, then it will correspond, this number will become 0, and will be a positive contribution to the energy. Also, if a pair of orthologs, say these two guys, they are both in a spin value equal to 2, it also decreases the energy of the system. So, of course, this is a random assignment at the beginning. So here, you can imagine that the energy wouldn't be that low. But the essential idea behind this mathematics is like the energy function is looking for some dense region in the networks, as well as try to align them across the species. So the ground state energy of this toy model is something like this one. So at the end, there are 3 modules emerge. So this is labeled by module number 1, number 2, and number 4. So one thing interesting is that we see 2 types of modules. One is this, like this module 4, we call it a conservative module, in the sense they consist of nodes from species A as well as B, and there are orthogonal pairs between them. So on the other hand, there are some modules which are specific to, say, this guy only contains genes from A, and this guy only contains genes from B. One tiny little point I want to point out is, at the beginning, we start with Q equal to 4. We have assigned 4 different values to the spin. But at the end of the day, it emerges only number 3 modules will emerge in this case. So it's possible. So just a little bit detail of the algorithm or the method we outlined. So I explained the use of this energy function, H. The input of the algorithm is basically two types of input. One is the orthogonal pairs between the species, and the others are the transcriptome of different species. So in the analysis I will show you is based on the transcriptome data from More and ENCODE project. So they are the worm-5 transcriptome across different developmental stages, and also the human transcriptome across different cell lines. So we use this transcriptome to construct co-expression network and arrive at the energy function. The way we try to get the ground state energy is by using stimulated annealing here. So at the beginning, we assign spin value randomly to the nodes, and then with a pretty high temperature. Then we let the system evolve according to the energy function, and wait until the system reaches equilibrium. So we then lower the temperature gradually, and then let the system settle down in some low energy state. So here I just show again some typical outputs of this algorithm, which are kind of Venn diagram-like structure with genes shared, gene from one species, gene from another species. They have orthologs, or they have some species-specific genes. So to make my life easier, I actually simplify the energy function a little bit. So there are actually a couple of more subtle points here I want to point out. One is the sign of the network is actually quite important. So because the genes can be positively correlated, meaning they are always together. Genes might be negatively related. So actually, one guy is here, the other is not here. So the intuition is positively correlated pair of genes are likely to be in the same module. But if they are negatively correlated, we should assign them to different modules. So the other is, in reality, the orthologs are not always one-to-one. So in many cases, we see many-to-many ortholog pairs. So we also have a simple way to do a normalization here. So you can imagine this energy function can be easily generalized to more species, say 3 or even more. So I just try to show you a typical one of this algorithm. So here, each trajectory is basically correspond to one of our algorithm. So at the beginning, we start with a pretty a high temperature, so the energy is high, and then this is the flipping rate. Flipping rate means that when I swipe through the whole network again, how many spin value got flipped. So at the beginning, both numbers are pretty high. So at the beginning, basically spin are flipping crazy. So everything got flipped at the beginning. But when we lower the system, and then so everything, the energy is decreasing, and the flipping rate reach a very low value. So we stop the algorithm by when the flipping rate is lower than a certain threshold. So of course, the labels, the spin value are just labels. So the same cluster, if I run it multiple times, it can be labeled using different names. So the way to really represent the data is use a kind of coappearance matrix to represent it. So here the matrix, the heat map basically saying that if I run the algorithm multiple times, how many times I got two nodes appear in the same module, in the same cluster of modules. So he did this run using the worm expression network together with the human expression networks. So here the clustering are sorted individually, but so if you look at this plot, basically to represent the cluster you can see in the worm along the diagonal. Look at this plot, the cluster of human along the diagonals. But of particular interest are some cluster like this one, the yellow guys here. So this one actually form a cluster in worm, this one form a cluster in human. But when you look at the off-diagonal position, these two clusters actually heavily overlap in the sense that this set of worm gene and this set of human genes, they actually appear very often in the modules. So I would, so we call this more conservative modules. So without surprise, when you look at how many genes are there, actually they share quite a large number of orthologs. So there are some other genes which, some other cluster which are we call specific. So say this guy is a worm modules, but when you look at the off-diagonal element, there's no human counterparts related to them. So this is the worm-specific guy, and this is the human-specific guy. So we do some standard goal-term enrichment analysis using for the conservative modules. So without surprise, also see that they share a lot of common goal terms. So both in molecular function or even biological process. So this is just a picture of the goal terms. So the size of the circle basically represent the p-values. So as expected, we find some big circle here, which is the ribosome structure, RNA binding, and also standard cellular metabolisms. So it looks like we are doing something not far away from reality. So this kind of conserved structure across organisms called cluster together. So we've also checked something similar for the specific modules, but here it's harder to make a nice story here. But in the case of worm, we got something like the flagella multiplicity genes, and the human, we got some signal transduction cascade. One possible thing I'm coming up is because of in the more encoded transcriptome, they're using some special cell lines to do that. The transcriptome are for some special cancer cell lines. So it might be the possible that this kind of structure pop up. So we also did similar experiment using worm and fly to arrive the same thing. So this is also the same heat map we are trying to show. So I guess the story is quite consistent using this worm fly and worm human simulation. So I want to, so as I said, this is an ongoing analysis. I kind of want to make a quick summary and outline something we are currently working on. So I propose this as a unified framework to do a clustering across two species. So once we have the cluster, of course, we want to kind of to use the cluster to annotate stuff. So one thing is to annotate the protein coding genes, but more interesting, the thing we are currently working on is to annotate the non-coding RNA. So we can map this non-coding RNA across from different species to this cross-species cluster. So I guess the interesting story is we are looking for some, we call it analogous non-coding RNA pairs. So maybe these pieces of non-coding RNA in worm correspond to some other pieces of non-coding RNA in fly, for instance. So even better, maybe they really, we can align it looking at the sequence, maybe they're not just analogous, but also orthogonal. So for future work, this kind of, this formalism can be extended to some more specific applications. Say, if you have the expression profile of human brain and a mouse brain, we can do a comparison like this one and look for some human-specific modules. Why humans are so smart or mouse are not so smart. And also we can do it apply in a bacterial setting and look for some environmental-specific modules. So to quick, I want to just acknowledge my lab mates. So I want to thank my PI, Mark Gerstein, and some of my colleagues, especially Daifeng Wang. He did a lot of upstream data collection for me. So of course, I want to thank the ENCODE and More ENCODE Consortium. So thank you.
Wonderful. Questions?
Yes. So the last thing you mentioned about using a mouse and human brain sounds very interesting, and I think more general human-mouse comparisons would be very valuable in learning which cases mouse models might be most effective if we could do a functional comparison between them, not only in the brain, but in many other systems. So to make that sound like a question, are you planning to do more than the brain in comparing mouse and human?
Am I planning to do on the brain?
To do more than the brain. Do human metabolism and mouse metabolism, do liver samples to see which aspects might be poorly modeled in a mouse in particular.
Yeah, I guess the method is very general. It can be applied to any setting. The reason I bring up the brain is like we got access to some brain span data, the brain span data, so that I brought it up. So I can definitely do it in liver or whatever.
So I'm a bit surprised that you're applying these methods freely across such distantly related species. The problem is that when you have these small numbers, say, of orthologs between human and worm, it means that you are not looking at most genes because there are no clear orthologs. Did you optimize this system in, say, the 12 fly data from mod encode, before going to distant lineages?
Did I optimize the system before what?
Before you went to distant lineages like human, worm, fly, did you optimize the system in closely related species? For example, study the 12 flies from mod encode where orthologs are easy, validate the system, then try to compare fly and worm.
I perfectly agree that worm fly or worm human, they are quite distant away. So. I guess in terms of optimization, I'm not sure whether it's, I guess the optimization have to be done in every different setting. So it's like, it's not like saying if I do it in worm, in a two co-species, I can easily apply it to another distant thing. I guess we need to do it again.
Great. And the last question is, so you mentioned at the end non-coding RNAs. As of course you are aware, ENCODE shows that 35% of human RNAs are absent in mouse, which kind of constrains your pool severely. So why do you think the system will be useful for non-coding RNAs, and what do you mean by analogous non-coding RNAs? I have not heard that term before.
No, okay, I invent this term. So I guess the nice thing about this cluster is they are cross species. So co and co, they are doing some function. So I guess if I have a cluster of protein coding genes which are responsible for the same function across species, I guess it's like a bridge so that we can link the non-coding RNA together. So it's like I can identify non-coding RNA in two species performing similar function. I think that's the term I use, analogous. That's defined this way. But yeah.
Wonderful. Thank you. And let's thank all the speakers for a great session.